Okay, so hopefully now you had a little idea of the genomic files that we have in the cluster. So the locations there tell you what organisms we have, and what I want to zoom in on now is both the genome reference files that we have available and the reads that you get out of the sequencer. They will come in specific formats. And these formats of files are called Fasta and Fastq. We usually say Fasta, but in theory, it's, if you want to be strict about it, it's Fasta. All right, so what are these files and how should you read them? So these sequencing files are used for the same thing. They have a lot of commonalities. So the main thing is they both store biological sequence information. And they also store the metadata for a given sequence. For example, the name of the organism, the name of the cell line you extracted it from, and the particular genomic location that sequence came from are all part of the metadata that can be stored in these files. They are both text-based files, and they serve different purposes. So we're going to go through each of them independently now and examine the differences and why two different formats are necessary. So first of all, the Fasta format. The Fasta format actually exists for a long time, since 1988, and it's a legacy from a sequence search algorithm that had the same name, which is very similar to BLAST that many of you may have heard or even used. And since then, this format has become the universal standard for storing basic sequence information, and it can be used to store sequences of proteins or nucleic acids. So the main files you will encounter that are Fasta files, they're usually labeled with .fa or .fasta endings. And it's a simple text format, and it's used to allow for us to easily manipulate and store these biological sequences. And what you will usually find it storing is reference genome files, but it doesn't need to be used only for that. It can be used to store any sequence information. You can make your own Fasta file. You will use it for input in several different programs that require it. So this is what it looks like. So there's a couple of standards that you should take note of here. So basically, every Fasta file will have first a line that starts with a forward caret and then is followed by the sequence name. So this needs to be a unique name for this sequence in that file, but it doesn't need to be a unique name in the universe. So it's only that you don't have two different sequences in the same file with the same name. And it doesn't need to have only the short name. It can contain a lot more information. And in fact, some Fasta files used as input for a lot of software have strict formats of which kind of information you should have there and how should it be written. Okay, then the next lines are all, they don't have any caret at the start. The caret is only on the name line. And the next lines will hold the actual sequence. So this is a stretch of the chromosome 1 that I took of mouse, chromosome 1, and it'll just go on forever until the sequence is over. It can be used to store the full chromosome or to store just a piece of it, and it should be labeled appropriately. And a Fasta file doesn't need to contain only one sequence. It can actually contain more. So in fact, you can have this file where soon after the sequence for the first ends, you have again a caret, a name, and on the next lines, a sequence for that second biological read or chromosome. Okay. So what about the Fastq format? So the Fastq format is developed more recently. It was originally created to store information from Sanger sequencing, and it has a little more storage capability than the Fasta, because in the Fasta you can store the name and the sequence information, and that's it. But the Fastq format, which you will usually find with these terminations of .fq or .fastq, is again a text format to store biological sequences, but it also stores the quality scores. Sorry, I went too far. I wanted to point it. But it also is used to store the quality scores for each base of those same sequences. And it's a very common format to have any kind of sequencing read information. So how does it look like? It looks like this. So you have the structure here with 4 different lines. It can actually be more, but I'll get to it later. So 4 different lines. The first line always starts with an @ character. And right after, immediately after the @ character, you have the read name or the sequence name. So this is the name here, and I'll break it down for you what this name means later. Then the second line is the actual sequence. So far, it's very similar to the Fasta format. It has a starting character, it has a sequence name, and then it has the sequence itself. But the third line of the Fastq format has this plus symbol, and it's used as a separator between the sequence and the quality lines. It was created just to separate the two and signal the end of the sequencing and the beginning of the quality. Sometimes you might see the name information for the read can be repeated here, but this is not common nowadays because we use very large Fastq files, and any kind of space matters. So when you have millions of reads, if you have millions of names, and this is a pretty long name, repeated twice, it's useless information and it'll just take up more space in your storage. So we don't want that. So more commonly, you will find this line empty. And then you finally have a quality score line, which is encoded, and I will talk about it later, but basically it'll have the same length as the sequence information line. And each of these characters encodes a quality score for each of the bases, the corresponding bases on the sequence. And like a Fasta file as well, the Fastq can store one or multiple sequences. So here is a longer Fastq file where you now have 2 more blocks of sequences repeated. So a second read here and a third here at the bottom. And the same structure is maintained for all of them. So you know where one of them starts because they have a similar structure. And a caution note that I had mentioned before is that I'm showing you here a 4-line Fastq file where you only have 1 line for the sequence and 1 line for the quality. But this is actually not a requirement of the format. It's more usually what you will find, but in theory, if you remember the Fasta file, the sequencing line could span multiple lines. The sequencing information could span multiple lines, and that same thing can happen in a Fastq file, but it makes life harder to have it like that, and most of the sequencing information we work with now are very short reads, so we can actually contain them in 1 line. So that's how you will usually encounter it. Okay, so what about these read names? They're pretty long and they contain a lot of information about the read and as an identifier, and it's composed of a series of strings separated by these colons. So this is the Illumina read name format I'm talking about. Of course, you can have different names in Fastqs from different software. So this is just the Illumina output that's usually like this. But what do they mean? So they each give an information about, first of all, the unique instrument ID that you used for the sequencing run. So this is a NextSeq instrument, NextSeq 500, and the number of the machine is 602. So the second information is the run ID in that system that you use to sequence. And the third information is the ID or the barcode for the flow cell that you use in that sequencing run. So there is a lot of information here that can be used to trace back where this sequencing came from, which machine did it, which run did it, which day was this run done in, if you have the logs for your system. And it can be very useful to track down errors or problems and do a lot of debugging. So then follows the address of the read. So basically the flow cell is a physical chip, right? It's a physical slide where each sequence, each fragment of DNA that you used in your, that you had in your library, hybridized and was read by the instrument. So this is the actual address for each of your reads. And you will have these precise coordinates encoded as the read name. It'll give it a unique ID. And then you can have some optional information. You can notice that it's separated by a space, and there's extra information here, which will tell you, for example, if this read is a member of a pair, since you can have a single end reading on your sequencing run, or mate pair reading or paired end, which we'll go into later. So this will tell you which member of the pair is this read, some other information about filtering, and if that thing will stop, and the barcode information that you use to label all of the sequences from a given sample. So this barcode here is specific to this sample, and multiple reads in your file will have the same barcode information. So now you know where it's stored. So, like I said, there's many reasons all this information can be useful, where you can do quality control or you can do detective work. For example, if you have a file that you downloaded from somewhere and you have multiple sequencing runs in it and some of them don't look so good and the mapping quality is not great, you can do some detective work and figure out, would they all sequence at the same time? Is it the same library? Should I expect batch effects? That's the most common way you will use this for. Illumina and their technicians can use it to go even further and see if there's any problems that happen during the sequencing run and do a more thorough quality control of anything that might have happened. Okay, so that's all the information in the read name for Illumina runs. And last, let's go through the quality line. So the quality line are characters that are actually encoding the FRED quality score. So what is the FRED quality score? FRED quality score is something that was created for Sanger sequencing runs as well. And it basically is telling you what's the probability that you call the correct base. So don't be intimidated by this formula. It's actually super simple. It has a straightforward mean. So the sequencing instrument is making a call for each of these bases, and it's reading the information that it got from the visual call and saying, what's the most probable base that was read here in this point, and it decides it's a G. So it's telling you in the quality line, what is the confidence that it has in that call. So you are familiar with Sanger sequencing, you can have peaks of different heights, and those peaks of different heights for each of the bases will tell, the higher one will have a more probability and the instrument will make a call for that base. But that call can be either very confident when you don't have a lot of signal from the other bases in that spot, or that call can be not so great when you have kind of a noisy read in that one nucleotide sequence. So basically, if you have have a probability of calling that base incorrectly of 1 in 10. So if you plug it into this formula, log 10 of 0.1 will be -1, and multiplied by -10, you will get 10. And that is your Phred quality score. And it signifies that you have a base call accuracy of 90%. So usually we consider a read to be a very good call if it has a Phred quality score of 30 or higher, because you can see that it dramatically increases your chance of having made the correct call. It may seem very strict to consider 90 a low quality, but if you think about how each of these has a quality score, having multiple bases of 90% accuracy can actually be pretty harmful when you're trying to find that tiny short sequence in the whole genome. Where did it come from? With a poor quality, and maybe you have the wrong sequence to begin with. Okay, so finally, I'm showing you here the Phred quality score has 2 characters. It can vary from 10 to whatever, but here it's encoded as just 1 base. And that is because they use this system of ASCII character encoding. And you can see that A actually corresponds to a Phred quality score of 32, and E corresponds to a Phred quality score of 36. So that's how you store the information in a minimum number of characters. And that's for this part. So now for us to continue the tutorial, you guys can go back to the, I can't go access my top here. Okay, so you guys can continue now on your own and go through this second link here of Fasta and Fastq files. So you can see what an actual Fasta and Fastq files look like. And you should find them all in the locations that we put here. And if you have any questions, please ask.
Okay, I guess 10 more minutes for this?
Yep, 10 more minutes.
10 more minutes, and we're going to start 11:15. Okay?
Yep.
Good. You can start now.